want to talk about cycles at first. And cycles are pervasive across all physical and biological systems. Cycles as significant as the rotation of a spinning and tilted Earth around the sun entrain billions and billions of plants and animals across the entire planet into various types of cycles that vary in duration, that vary in complexity. Cycles occur at all levels of complexity, in fact, from the biochemical level to the cellular level to the individual level and even to the population and species and even to the community level. Some cycles that are entrained basically because of the spinning Earth around the sun entrain light-dark cycles. And billions of animals are entrained into a circadian cycle. We're all familiar with many types of different circadian rhythms that drive so much of the biology we see. What I'm going to talk about today is that these cycles, such as the seasonal cycle, from summer to spring to fall to winter, or in the tropics, these precipitation or wet, dry cycles, similarly drive enormous amounts of the biology that we see in so many plants and animals. But what I'm going to argue is that we've missed the boat in so many respects in terms of studying cycles as a fundamental aspect of the biology of most animals and even many plants. We've caught it in some areas. And I would argue that in the physiological literature, there's been a real strong attention to different types of cycles. But in the ecological and evolutionary and even the conservation sciences, we've ignored cycles for the most part. So I did a review of when, during the annual cycle, research had been conducted on amphibians, reptiles, birds, and mammals. And I'll just present three of them here, amphibians, birds, and mammals. What I found was that over this time frame, that over 96% of the research on amphibians had been conducted just during the breeding season. And most of us in this room, since most of us are natural historians, understand that for amphibians, that's a very short peak during that period of annual cycle. But that's, because it's easy, because that's when we can study them, that's when most of the research is done, ignoring most of the rest of that annual cycle, let alone the actual life cycle, which I'm really not going to talk about because that's a whole other level of complexity. For birds, 75% of the research had been conducted just during the breeding season. Mammals, a little better, 67%. Reptiles were about 70%. If we drill down a little bit more and look at passerine songbirds, again, 75% during the breeding season, 12% during migration, which is non-breeding season, 10% during the winter, which is another period of the non-breeding season, and then 3% include what I call seasonal interactions. That is how these different periods of the annual cycle interact, which I would argue is exactly what we should be focusing on with all of our biology, trying to understand these interactions, which are probably the norm. That should be the null. That really seasonal actions drive everything, that only things that happen during the breeding season are responsible for most of the variation we see in ecology and evolution of a variety of traits. But I would say that these interactions are probably the norm, but that's not what we've been studying. And there's lots of reasons why I think this occurs, this extreme bias in when we've conducted our research. So there's a variety of reasons, some of which are the fact that so much of the research is already conducted in temperate areas, and that there's a temperate bias in when research is done by academic institutions during the summers, which happens to also be the breeding season. But there are also technological barriers which have caused this problem to occur. Then the fact that we can't track most animals throughout their annual cycle or throughout their life cycle. It doesn't matter if it's a fish and you're talking about a larval fish moving to an adult stage. It's impossible for us to track these things. Or it's a jellyfish, or it's an amphibian, or it's a bird. We are limited in our tracking technology, significantly limited in understanding movement dynamics and how these things influence the biology of these animals. Now, I'm not going to talk about that in a lot of detail. That's a whole other talk that I could spend hours on, but I'm not going to focus on that. I'm going to focus on these seasonal interactions and how they're really fundamental to the biology of these birds. This is the American redstart. It's a species I've been studying for over 25 years now, and this is its basic annual cycle. What I want to focus on is that these birds first arrive in the breeding grounds, if you notice the red, in between May and June, early May. Males and females pair up. They're on breeding territories anywhere from Louisiana all the way up to Alberta and Alaska and Newfoundland. They have a really wide breeding range. And then breeding ceases, usually in July. They're obligate single-brooded species. Males and females split. Fledglings take off after that parental care period's over. And they go through a full body molt. That doesn't all happen at once, thank goodness. And then they go on a migration. That migration takes about 2-3 weeks, we think. And then they're on the non-breeding grounds for the entire annual cycle, which is about 6 to 8 months, depending upon where they are and their sex and age. But they're on the non-breeding grounds for the majority of the annual cycle. And what my basic hypothesis is that I'm going to test today, to show you today, is that these periods of the annual cycle interact. That if you just study this breeding period, you're going to miss most of those drivers of the underlying variation of much of the biology of these birds. So what I'm arguing is that things that happen during this non-breeding period drive things during migration and during reproduction. So what I'm going to tell you the answer right now. What I'm going to show you is that most of the reproduction that we see during the breeding grounds is actually driven by things in the tropics, thousands of kilometers to the south. We have really good evidence for this now for multiple species. I'm going to show you some evidence for just 2. And what's driving that, what the underlying driver is either habitat suitability or the surrogate for habitat suitability, which is precipitation across multiple years. So it's essentially moisture. It's a moisture gradient that either occurs by different habitats or across years, wet and dry years, that drives underlying food. It's all about the food that's driven by insects or fruit that's driven by precipitation. So seasonal interactions are this notion that we came up with theoretically, but it's an idea that's been in the literature since Darwin. I've actually found Darwin notes that actually talk about this idea of seasonal interactions. These are circumstances within one period or season of the annual cycle that influence events in a subsequent period. And they occur, we've sort of put them into ecological and evolutionary categories. They occur at the individual level in the form of what we call carryover effects. And these are non-fatal effects, where an individual is somehow compromised, either physically but doesn't die, or by time. So they leave their wintering territories or their breeding territories a little bit later, and that somehow carries over into the next or subsequent period, either migration or the wintering grounds, to influence some other trait in that animal. They can occur at the population level in the form of density-dependent effects. You might imagine a year of really high reproductive success, where you've got a lot more recruitment into the population on the wintering grounds that subsequent winter. You have density-dependent effects that occur. Fundamental Fretwell ideas here. Just the number of territories, the number of individuals have a limited number of resources, so they're forced to spread out across areas or compete more for limited food resources. That's a population-level effect. And that can occur, if you think about it, and think about your own organisms as I'm talking, because all these things really relate to multiple types of species. But these population-level effects can occur in multiple directions and in multiple ways. I'm going to focus on winter to summer, but you could think about this in any direction. And they can also be maternal effects. There's all kinds of cool ways to think about these interactions. And they can also occur in an evolutionary perspective across generations. The color that we see in birds or the size of a morphological trait is not driven just by things that happen on the breeding grounds. Sexual selection, it's all been about breeding season. But when you start to think about sexual selection, those same traits, whether it's a long tail in a bird or a color, can be selected against during the non-breeding season. And animals that want to survive into that next generation, of course, have to compensate in order to survive. So sexual selection, the traits that we see, even just for something that's so focused on a breeding ground, are selected for and against throughout the annual cycle. And if we really want to understand the evolution of these traits, we need to think about them as these animals are exposed to these different selective factors throughout their annual cycle. And ultimately, I admit, throughout their life cycle. But that's not going to be in my lifetime, I don't think. Maybe, I hope. So today I'm going to really focus on this one simple question that I'm going to show you. What's the role of moisture and food during the non-breeding season in driving bird performance both within the non-breeding season? So we really have to focus on that first, because for the longest time we really haven't understood the influence of climate on bird biology. So it's a constant frustration for me, because everyone wants to understand the role of climate change, you can't look at climate change unless you know the fundamental biology of how climate influences the biology of the animal, whether that's the physiology or the ecology or whatever it happens to be. Then we have confidence in projecting forward. So I'll be talking first about within-season effects, and then these between-season effects through carryover effects. And I'll do that with two species. One, the American redstart that we've been studying actually throughout its entire range, but primarily in Jamaica, where we have a 25-year study. We've got a crew of about 12 people right there right now working on various aspects of this species. And then the Kirtland's warbler, which we've been studying for the last 10 years, both here in the Bahamas with collaborators, these two old guys that Eric keeps pointing to, Joe Wonderly and Dave Ewart. He's the one. This guy over here is the one. I have to pick out Stedman at some point, too. Those are the three guys we're picking on. Is that right, Eric? And so we've been studying this species both on the breeding grounds as well as here, directly and indirectly, in the Bahamas. The general idea is fairly simple. And again, we can think about this as being driven by rainfall as the driver or across moisture gradients across habitat suitability types. And the idea is that rainfall influences primary productivity and or territory microclimate and vegetation structure and influences food. In the case of redstarts, that food is entirely insects. In the case of Kirtland's warblers, it's both fruit and insects. And that can either change the carrying capacity of the site or the year and influence bird density, or that can influence body condition of the birds directly, or that bird density can influence body condition through negative density-dependent effects. This influences when birds depart for the breeding grounds, and I'll show you those data. We were the first people to really show that that's a really important mechanism here. And that influences the probability of survival during migration or the probability of survival back on breeding grounds and how well these birds do during reproduction, how many young they actually produce. That's what I'm going to argue or show you data for today. This is the annual cycle of the American redstart, which I've already described to you. Redstarts during the non-breeding season are a wonderful species to study in many respects. They consume only arthropods, so that's a complex thing in itself, but once you add in fruit, then really understanding and quantifying the underlying food base becomes a little bit more complex. But we spent a lot of time doing all sorts of techniques. I'll be happy to talk to you about these. Everything from genetics of the fecals to stomach washes, doing all kinds of things to really nail down the food. Males and females on the wintering grounds hold individual and exclusive territories for the entire winter, for the most part. There is a floating class as well that we're starting to understand a bit more. So it's a fairly complex social system, but we've been studying it for a while, and it makes it easy to capture birds multiple times over the winter period. They're easily sexed and aged, both visibly and in the hand. And again, as I mentioned, individuals can be recaptured multiple times, so we can do all sorts of physicals on these birds to assess their condition over the winter period. The research, as I mentioned, is primarily in Jamaica. This is the general range of the species. It's fairly widely distributed. We work in the southwestern portion of Jamaica between Sav Lamar and Black River in a little area known as White House. We study them primarily in two habitat types, but they actually occur in many habitat types. We've chosen two for these intense studies. So this is black mangrove swamp, and we're in this habitat when the birds first arrive, usually in the fall or early January, early winter, and the water level can be anywhere from 1 to 2 meters in depth. This habitat stays fairly moist as that tropical dry season progresses. Unlike the scrub habitat, which neighbors the black mangrove habitat, and this habitat actually is female biased. This habitat is fairly lush when the birds first arrive. You can see on the left, but as the tropical dry season hits in, say, March, this same site, same spot, this is a deciduous forest, so these trees lose most of their leaves, and it becomes quite the harsh environment for the birds there. We capture the birds and measure them and band them. We do all kinds of things, weigh them. We've looked at corticosterone. We've done enormous numbers of things on these birds to assess their condition. And we then recapture them and weigh them for mass change and do a variety of things to see how they're doing in these habitats or in different years. And then we also recite them every 3 days to figure out when they leave their territories to head north. And we do this every year. We've got 20 years of data on this. And we've manipulated this system as well. And then we recapture and recite them in multiple years because they're all individually color banded. So we get pretty good data on capture, recapture, survivorship analysis, and measure food for arthropod sweeping. Now I'm just going to tell you the answer here, but basically performance depends on habitat occupancy or the severity of the drought in a particular year. In general, in black mangrove, food availability stays high over the winter period compared to second growth and birds in that habitat, regardless if you're male or you're female or you're young or you're old. So it's regardless of your age or sex class. If you're in black mangrove, you're going to do pretty well. Your body mass is going to be maintained, your spring departure is early, and your annual survival is high. In the scrub habitat, food availability tends to decline over the winter period. Similarly, birds in that habitat, regardless if you're male or you're female or you're young or old, your body mass declines, your spring departure is late, and your annual survival is low. You're probably saying, Pete, this is just a correlation. Well, I don't have time to show you, but we've done removal experiments in the black mangrove, upgraded birds from scrub, and we can manipulate all these traits in these same birds with birds that are known, relative to controls, that are known control birds in scrub habitat. And again, you can also look at this across years and look at wet versus dry years. On the x-axis is corrected body mass. Think of this as basically condition. This would be bird 0 would be a bird that maintains its condition over the entire winter period. -8 would be a bird that lost 8/10 of its body mass. The y-axis is spring departure. Red is scrub. Blue is mangrove. So again, there's a clear habitat effect here. But in general, birds in better condition in wet years leave earlier. And the interesting thing is that because all these birds are color banded and we're tracking these birds in a longitudinal way across years, we can look at how the same individual birds change over multiple years relative to the conditions they're exposed to in these wet versus dry conditions. And what we find is that depending upon the amount of rainfall, the same individual might leave 4 or 5 days later if it's a drier year, regardless of the habitat. Or if it's a really wet year, they'll leave 4 or 5 days earlier, which has never been known before. Most people have thought that the onset of migration is an endogenous driver. And what we're showing now is that is not endogenous. It may be initially, but there is definitely an environmental component that explains some of that variation. We've done this with multiple species now. So let's start asking, those are sort of the within-season traits that I described to you. Let's ask this next question, whether or not these things carry over and influence some things during the breeding season. So ask the question, does winter precipitation predict breeding ground arrival times and first egg dates in breeding redstarts? This is up in the Queen's University Biological Field Station where we work up in Canada. And what we found was that the rainfall in Jamaica, and we don't know for a fact that these redstarts that we study in Jamaica actually go to this site in Ontario. We don't have very good connectivity. We know, using a variety of stable isotopes, that they're probably linked, but the direct links are not known. We don't see the same birds in both places. But what I can show you is in that top graph, these are females, first egg dates. First egg dates are much later, significantly later, in dry years relative to wet years. It's an R-square of 0.769. We don't see that in ecology very often. It's a pretty important link between what's going on in the winter with precipitation and first egg dates. We first found this back in the days when people were really talking about how birds are arriving earlier on the breeding grounds with warming temperatures, and I just was holding up my hands and thinking, guys, you're missing the boat. It's the tropics. Things are happening in the tropics. It's precipitation on the wintering grounds that's determining when these birds leave. It's not just temperature that's changing on breeding areas. You've got to think bigger. And so looking at males and first arrival dates, same sort of pattern. Not quite as strong, but still a significant relationship between winter precipitation and first arrival dates in males. So let's then ask that next question. Does non-breeding habitat occupancy influence the number of young that are fledged? In other words, are there traits other than habitat availability, breeding predation, but things that happen prior to when these birds arrive that determine the number of young fledged? The answer is yes. So to get at this, because these birds are so damn small and we haven't been able to really track them, which is the case for most organisms, I use something called stable isotopes, carbon-stable isotopes that vary as well during this, or across this wet to dry moisture gradient and across wet and dry years. And it's great because, as we all know, we are what we eat. And when you eat things in a dry environment, you tend to incorporate isotopes that are more enriched in delta C13. And when you eat things, insects or plants, in a wet environment, you tend to, your blood, your tissues, your nails, your hair, whatever it happens to be, or your feathers, tend to become more depleted in delta C13. And depending upon the tissue that you look at, that tissue will carry that signature for multiple weeks or even for a year. So what I did was I sampled birds. I demonstrated that this was the case in black mangrove and second growth scrub in multiple years, but this is just a comparison across 2 years. And I sampled birds as they arrived on breeding areas and found that that same pattern occurred. Birds that arrived back early, I don't know what habitat they were coming back from, but they tended to have signatures that were more depleted, similar to animals coming from a moister environment, versus birds coming back later that were coming back from habitats that were more depleted or more enriched or more drier environments. We've shown this now in multiple species, multiple sites, even in shorebirds. It's actually been replicated many times now. And then what we do is we follow these same birds once we have their winter habitat signature, and we can look at the different factors that drive their reproduction. And so for American redstarts, x-axis is wet or delta C signatures that are coming from a moist environment out to dry. And you can look at the proportion of offspring sired, so actually look at how much hanky-panky is going on out there by individuals, probability of polygyny, individuals that have more than one female, or the total offspring fledged. And what you find is that birds coming from wet habitat fledge about 1.5 to 2 young more than birds coming from dry habitats. All right, let's shift gears a little bit. It's enough about the redstart. Let's talk about the Kirtland's warbler. Kirtland's warbler, most of you are probably familiar with them. They're a small migratory bird. They're endangered. They were down to 200 singing males back in the 1970s, thanks to a lot of work that was going on only on the breeding grounds, removal of cowbirds, habitat management. The population is now up to about 1,800 singing males. It's not the smartest bird in the drawer, let me put it that way. They're highly specialized on both ends of the spectrum. They're really easy to catch. They are big and they're yellow, and they're pretty dumb. They're ground nesting species. They eat both fruits and insects. They're a fun bird to work on, though. Their life cycle is a little different. They spend quite a bit of time on the breeding grounds, about 4 months of the year, and about 6 months of the year, maybe 7 months on the non-breeding grounds. Spring migration, as you'll see, we've nailed, thanks to a variety of different approaches, but it's about 2 weeks, and fall migration is also equally short. Here in the Bahamas and other places where they winter, as Joe and Dave have shown, they're highly specialized on a scrub-type habitat. They're very hard to observe. Their breeding grounds are also very specialized in this jack pine habitat, but it's not just jack pine. It has to be the right successional stage of jack pine. Jack pine is a fire-dependent species. They show pretty strong within and between year site fidelity. Very strong on the breeding grounds, somewhat strong on the winter grounds, not quite as strong. And their winter range has long been thought to be primarily in the Bahamas, but we'll confirm that with some other work in just a bit. I've had a variety of people work on this, but Sarah Rockwell has contributed the most to this research. She asked 3 main questions. How does winter rainfall predict spring arrival dates, reproductive success, and annual survival? Studying the birds from a breeding ground perspective. She's done some work in the Bahamas, but most of her work was actually up in Michigan. She banded over 380 Kirtlands, recorded 483 arrival dates for 306 different individual males across multiple years, and she found over 270 nests during her time as a dissertation student. What did she find? This is March rainfall in the Bahamas, so we can really, because we know so much about the winter distribution of this bird, we can do a pretty good job of isolating where we need to get climatic data. So we can look at March rainfall in the Bahamas relative to arrival date. What's interesting here is that for ASY males, those are older, after second year males, or older males, think of that as an older, SY as a second year male or young male, there's pretty much there's no relationship at all. These birds, once they're fixed into a certain pattern, sometimes they're not that flexible, at least with respect to arrival date. SY males, however, show a really strong relationship in dry years coming back later than wet years when they come back earlier. What about number of young fledged? This was quite striking. March rainfall again, number of fledglings on the y-axis. Clearly, Kirtland's warblers raise fewer fledglings, fewer young, following dry winters, and in the same pattern across both age classes, older birds and younger birds. Younger birds typically fledge fewer young. But again, really tight relationship between precipitation in the Bahamas and the number of young that these animals produce. So there's a really strong linkage, regardless of the fact that we're talking about 2 very different places, separated by multiple kilometers. And what about annual survival? So it's important to look at these fundamental vital rates and how they are driven by these different factors. Again, really striking, survivorship is driven by winter rainfall. In really dry winters in the Bahamas, survivorship is significantly lower, around 55%, compared to wetter years when it goes up to about 65 to 70% survivorship. So we can actually now look at this and put it into an annual cycle or full annual cycle perspective to understand when during the annual cycle are these birds dying. So we know we have over summer survivorship for Kirtland's warblers in Michigan. Joe and Dave have collected data on over winter survivorship. And then we can sort of, by knowing those two things and knowing the annual survivorship, you can do some back-of-the-envelope algebra and figure out what the mortality is during spring migration. And what you find is that actually most of the mortality, the survivorship, goes down during spring and fall migration. We think most of the mortality is actually during spring, but we haven't separated it here in this case. But in general, 41% of the annual mortality occurs during this two months of migration. And in some other analyses we've done, what we've shown is that it's not just migration mortality. Again, there's an interaction. So depending upon the habitats the birds are coming from, this is from Redstart work and northern water thrush work, depending upon the habitats the birds are coming from or the severity of the winter, that determines the mortality during migration. So there's an interaction there again. That's a really important thing. But in general, we haven't known that much about migration. So we've continued to sort of study this period to try to figure it out. So in general, I hope I've convinced you, at least for the Kirtland's warbler and the Redstart, that winter rainfall is really an important driver. Determines when males and females arrive back to the breeding grounds. They fledge more young and offspring. They survive in higher rates when they're coming from wet habitats or after wet winters. And most of that mortality is during migration, but it's because of an interaction with the habitats or the severity of winter during the non-breeding season. We've actually shown this in northern water thrush and ovenbirds, but I don't have time to go into the same sort of results. But these patterns are becoming pervasive. These patterns are becoming commonplace. We're seeing these across multiple species and not just passerines as well. So let's look at this migration period just a little bit more with some recent work that we've just been getting into. This, for the most part, unless you can track birds with satellite transmitters, we haven't really known much about what happens during migration. It is a black box. Birds just disappear, they go off. We don't know much about stopover biology. We don't know much about the length of time. But given that most of the mortality occurs during this period, we really need to understand what's happening there in order to do the relevant conservation and understand the fundamental biology. So in recent years, there have been some really remarkable technological developments that allow the tracking of smaller and smaller animals. This is an example of light-level geolocation that we've been using. These are small tags that weigh 0.4 of a gram. It's the lightest ones that are available now. And they measure 24-hour light intensity. So you put them on the backs of these birds. Basically, it's a harness that goes around their legs. And you let the bird go, and you hold your breath. They cost about $180. These do. They vary in price. And then you've got to catch them the following year. So essentially, because survivorship's about 50%, you're basically throwing half of them away because you're not going to get those back. But they store the data, so there's no transmission up to a satellite or a cell phone tower. But you can then take these data, once you get them back, you catch the bird the next year, you download the data, you look at light intensity. And if you know light intensity, aside from the equinoxes, because the light intensity is the same, it's about 12-12, you can infer geographic position, plus or minus 150 to 200 kilometers. So it's not great, but it's a hell of a lot better than where we were. So Nate Cooper, one of my postdocs, we put these on about 60 birds, and we recovered 27 of them on the breeding grounds because they show high site fidelity, because we can recapture these birds, because they're kind of dumb. We were able to recapture 27 of these birds on the breeding grounds, and we got some pretty cool data. This is a paper that's in draft. We're about ready to submit it. Basically, we found that about the vast majority of these Kirtland's warblers up in Michigan did what we expected. And given what Joe and Dave would have predicted, they wintered primarily in the central Bahamas, but not entirely. You can see there's basically a lot of variation. This is the sort of a summation of all 27 of those birds, incorporating all that variation. But basically, we found 3 in western areas, 19 in central Bahamas, 3 in western Bahamas, 4 in eastern Bahamas, and 1 that probably went to Cuba. which is pretty interesting. But there's a lot of different things that you can learn from these data as well that we didn't know before, well, for the most part. In general, they departed the wintering grounds in late April, which Joe and Dave kind of knew from following color-banded birds. And it took them about 15.8 days to get, according to geolocation, to get from the breeding grounds, or from the wintering grounds to the breeding grounds. So Joe and Dave have been tracking color-banded birds on both ends of the spectrum, and their paper that came out a couple years ago calculated it as 15.8 days, which is pretty remarkable. So sometimes the old ways work. Departure date is the best predictor of arrival date, which was also not surprising, but it was great to have it confirmed here with the geolocator data. And each additional day spent on the wintering grounds meant 1.2 days spent on stopover, and it led to a 1-day delay in a spring arrival. And every day you delay, there's a cost to reproductive success, which is the mechanism that was I was alluding to earlier in those other studies. We also found there were three, after birds left the Bahamas, there were 3 primary stopover areas that no one really knew before, which is pretty interesting. Landfall in Florida in the spring turned out to be pretty important. And then a secondary site a little further up in southeastern Georgia, just before these birds crossed this other biological barrier, what we think is a barrier, the Appalachians. And then up in southern Ontario, they seem to stage here too for about 2 days. Each of these other sites is about 5 days, and then about 2 days here, just before reaching the breeding grounds. Now we've animated this, which also includes the fall migration. Let's see if this works. I'll talk you through it. So this is the departure of this mass of geolocated birds in April, moving up to Florida, and then staging in just before they cross the Appalachians. First crop of birds is moving across the Appalachians into northern Ohio and southern Ontario. They're starting to arrive in the breeding grounds. Now, pretty much breeding starts to commence at this point. They're there for a couple months. These birds will be multiple brooded, depending upon the year. They'll re-nest if first nests fail. But they stayed on the breeding grounds longer than we thought, and they moved further east to what appears to be another staging area, perhaps where they're molting. We don't know. This is new. And then they start migrating, but early October, I don't think people really recognize how late they actually stayed up there. There's a lot of variation here because we're near the equinox. But then they're arriving back in the Bahamas. What's interesting is they actually took a different departure point over here. And so what they're actually doing is a loop migration up here, back down over here. And their departure was primarily over in this region. So a very different departure from the United States than where they made landfall into the United States. So that was hot off the press, so I just wanted to show you those data. They're pretty cool. I hope I've convinced you that seasonal interactions driven by winter rainfall are really important. Winter ground effects can delay arrival and limit productivity and survival for not just redstarts and Kirtland's warblers, but many migratory species. And the bad news is that yesterday we kept on talking about sea level rise, which is obviously a big, big problem. But drying is a huge problem in the Caribbean more broadly. Nealon came out with a wonderful paper in PNAS in 2006, which predicted a 20 to 40% decline in precipitation over the next 20 to 40 to 50 years. And you were seeing this in the Bahamas. The drought data in the Bahamas are really convincing. The drought data in Cuba and Jamaica and throughout the Greater Antilles is really severe. We're seeing these effects in all sorts of ways. This drought, this importance of winter precipitation, is something that we also need to really study. If you aren't already, I'll be really interested to see what talks go into that today and tomorrow. But if we ultimately want to conserve species, there's a variety of things I would argue we have to do. We have to move more towards a full annual, if not a full life cycle approach to our biology, our conservation, certainly our outreach. And I think the Kirtland's warbler example is one I always point to as this wonderful example of where so much great training has happened throughout the annual cycle. And we need to do that with all of our research, regardless of what it is, and our policy. And I can't underestimate the importance and overstress the importance of technological advances in all that we do. I spend a lot of time working on trying to advance our ability to track animals and track movement because it's so fundamental to our ability to really understand that biology and do the conservation. It's essential. But ultimately, it's what we're going to learn from these devices, such as where these stopover areas are or when these birds are departing the U.S. and where that happens to be, that we can start to look at where to protect habitats, how we can prioritize, and then we can start removing the threats. And we can do wildlife management, but it's going to have to be throughout the annual cycle. Thanks very much.